Hi students, in the last class we discussed about the cytokines and the bioassay of the cytokines and the physiological functions of the cytokines, history of the cytokines. We discussed in the last class, right? And coming to the next topic is the ethylene, right? Ethylene, this is also one of the important phytohormone. Remember, the formula for the ethylene is the CH2 double bond CH2. CH2 double bond CH2. And this is a gaseous hormone. This is a gaseous hormone. See, this hormone available in gaseous state. So, where we can find this hormone? Especially fruits. Whatever the fruits are there, okay. We know about the biochemistry of fruit ripening process. We discussed so many times whatever the green color fruits are there, the green color fruits turns into the ripened fruits. That is nothing but biochemistry of fruit ripening process. Biochemistry of fruit ripening process. So during the fruit ripening process, whatever the hard parts are there in the fruits, whatever the hard parts are there, the hard parts are converted to the soft or smooth parts. Because of the action of some of the enzymes, especially hydrolysis are there, and the cell wall degrading enzymes are there. All these are act on the cell wall parts and the storage food material. Then the hard fruit is converted to the soft fruit. That's nothing but the green fruits are turns into the ripened fruits. So that is about that the best example for the role of the ethylene hormone. Here, ethylene hormone first invented by the Cousin. The scientist name is the Cousin, right? And later, activity of ethylene hormone, identified by the Berg and Thiemann. Berg and Thiemann, both the scientists, identifies the activity of the ethylene phytohormone. And see, ethylene is the derivative phytohormone. All phytohormones are derived from different substances, but this one is quite different from remaining phytohormones because this phytohormone is derived from the amino acid. Ethylene phytohormone is derived from the amino acid. Name of the amino acid is methionine. See, methionine is the source for the production of ethylene phytohormone, right? And the methionine is the sulfur-containing amino acid. The sulfur-containing amino acid involved in the production of the ethylene phytohormone. It forms one of the intermediate structure between the methionine and the ethylene. The structure name is aminocyclopropane carboxylic acid. Very simple. Aminocyclopropane carboxylic acid. This is the intermediate structure between that methionine and the ethylene. Again, this product is converted and finally produces the ethylene phytohormone. Right? That is about the methionine and the production of the methionine. See, bioassay. Bioassay, we can decide here based on the color of the lemon. See, green lemons are there. Green means these are not ripened. The color is the lemon color is the green color. Ripened lemons are there, yellow color. Whatever the ripened lemons are there, which are exhibit the yellow color. So based on the color, we can estimate which one is ripened and which one is unripened. Right? So here we are doing that bioassay based on the lemon coloring test. First of all, in a basket or in a bowl, we kept a few lemons and banana. These are the green. Green means these are not ripened. These are the green lemons and these are green bananas which are not ripened. Right? After few days, see the color of the lemons change into the yellow color. Once the lemon colors are changed into the yellow color, automatically whatever that ethylene phytohormone is produced here, that phytohormone also affect on the remaining green fruits. The universal phytohormone for the fruit ripening process for any fruit, that is the ethylene. That is the ethylene. Lemon produces the ethylene. Okay, when the ripening takes place, or lemon required for the ripening process, ethylene. Every fruit required for the ripening process, ethylene. Right? So here these fruits are produces the ethylene. That released ethylene again affect on the remaining green fruits. So once these are ripened, these are also ripened because here ethylene is produced. Again that ethylene is added on these fruits. Again these are ripened. Again any other things are there. Automatically here step by step the ethylene hormone is produced. The surrounding all green fruits are ripened. Right? That is about that. By a status based on the color, we can estimate the fruit ripening process. Then, if the green fruits are immediately ripened, then we can conclude there is a high concentration of ethylene is present. So, some of the fruits are slowly ripened. Okay, we are taking two bowls here. This is the one bowl and this is another bowl. First bowl, right? Here we kept 3 lemons. These are fully ripened lemons. And we kept here bananas. These bananas are immediately ripened because these are fully ripened. And we kept here some of the ripened. These are not fully ripened. These are just ripened lemons. And we kept here banana. Right? Maybe these are required 2 days or 3 days for the ripening process. These bananas are required for the ripening. The time period is here. Maybe 2 or 3 days are required. Because here these lemons are fully ripened. These bananas are immediately ripened. See, these are at intermediate stage. These are just ripened. So here, production of ethylene hormone is slow. Okay, the bananas also slowly ripen. Right? So here, this is one of the chemical name. Okay, this is one of the chemical substance we know. In the fruit markets, this is one of the interesting topic. We know about the fruit markets. So within the fruit markets, some of the chemical substances are available. These are also called battery. Okay, in our local areas, this is also called battery. That name is ethopon. Right? That ethopon releases the ethylene phytohormone. Ethylene. Okay, this is the chemical substance releases the ethylene. So presence of this substance, the fruits are rapidly ripened or immediately ripened. So this is one of the artificial ripening process. We know this substance also banned by the government because when the fruit sellers uses this substance for the fruit ripening purpose, that time some of the carcinogenic substances are produced. So presence of that carcinogenic substances, whatever the fruits we ate, which are ripened by these substances, by the ethopon substances, most of the consumers, there is a chance to get the cancer. See, that problem is avoided by the prevention of usage of ethopon substance. The ethopon substance, right? See, this one is also chemically called 2-chloroethyl phosphoric acid. That is the name of the substance, right? And see, ethopon, there is 2 types of effects are there. Positive effects are there and negative effects are there. Here, positive effects are flowering, right? Flowering in apple. And next is the fruit ripening process. We know this is the universal one, fruit ripening process. And in some plants, very specifically, induction of flowers. There is nothing but flowers production is promoted. And negative effects are there. Abscission of leaves and flowers. We know the leaf fall off, flower fall off is majorly done by the ethylene also. See, abscisic acid is the major one, but the ethylene also one of the growth inhibiting phytohormone. And the thinning of cotton fruit. See, the chemical value of cotton fruit is always depends on the thickness of the cotton fruit. So here, when you spray the ethylene hormone, here whatever the thickness is there, the thickness of the fruit is reduced. Then the chemical value of the that is about that the ethylene substance, positive and negative effects. See, this is the ethylene production process. This is the graph, right? The very first case, unripened fruit. Unripened fruit also having certain percentage of the ethylene phytohormone, because when the ethylene is there, that time ripening takes place. Here concentration is very low. Okay, ripening process, the concentration of ethylene levels are increased compared with the first stage, the second stage, the ethylene levels are increased, and the fully ripened stage. In that case, again the ethylene hormone, the levels are increased. At fourth stage, over-ripening process, very high concentration of ethylene phytohormone is present. Very high concentration of ethylene phytohormone is present. So, this is the production of ethylene phytohormone, right? This is the explanation based on the graph. This is the explanation based on the graph. And see the functions of ethylene phytohormones. Functions of ethylene phytohormones. Negative effects. We know abscission. See, whatever that attaching parts of the flowers are attached to the stem or twigs, leaves are attached to the stem or the twigs. In that case, see, between these two parts, one of that the layer is formed. The layer is called as abscission layer, right? So, abscission layer is formed. There is a leaf fall off, flower fall off, and the end stage fruit fall off will be takes place. That is one of the disadvantages to the farmer. So here negative effect of the ethylene abscission layer formation, right? Senescence of flowering, old age. See, when the flowers are formed, these are immediately fall off. That is nothing but senescence, right? By the abscission layer formation, right? And as soon as the positive effects are there, these are very peculiar. These are only seen in some plants, not in all plants. Flowering in pineapple plant, okay, specifically in pineapple plant, flowers production, okay, induced by the flower production done by the ethylene phytohormone. And as the flowering in mango plant, and internode elongation process, whatever that the stem contains that day. Okay, we know about the node parts. We know about the node parts. This is the node, this is the node, this one is the internode elongation which takes place. And root case formation. Okay, whatever the root is there, the root having that root case. See, within the maturation region, more number of root cases are there. The root case production done by the ethylene phytohormone. Feminism in cucumber. We know femaleness. More number of female flowers production takes place within the cucumber, and the break the dormancy. Whatever that seed dormancy is there, the seed dormancy is broken down by the ethylene phytohormone. Not in all seeds, okay, especially in some seeds. Basically, the seed dormancy is broken by the gibberellic acids. Remember, right? Here, peanut germination process. Peanuts germination is supported by the ethylene phytohormone, and potato sprouting. Okay, whatever that potato plant is, they sprouting. Some of the small growths are exhibited. The tuber exhibit that very small green color growths. That is nothing but sprouting. So potato sprouting is also promoted by the ethylene phytohormone, right? So these are the history and biosynthesis, and this is the bioessay, right? And these are the artificial ethylene phytohormone producing products, positive and negative effects, graph explanation for the production of ethylene phytohormone, and these are the positive and the negative physiological effects of the ethylene phytohormone. Right? Next we will discuss about the abscisic acid. Yeah, this triple response also related to the ethylene phytohormone. Sometimes asked in the bit, mostly NCERT not given clarity into the triple response, but remember this is one of the important area regarding to the ethylene phytohormone. Okay, ethylene exhibited a triple response. What is triple response? Right, ethylene exhibited or presence of ethylene, some of the seed shows the triple response. What is triple response? Remember, the very first one is the apical hook formation. Apical hook formation. Whatever the seeds are there, whatever the growing seeds are there, especially dicots, so which forms the hooks-like structures. When the seedling takes place, that time hook-like structures are produced, especially in dicots. Especially in the dicots. So during the dicot seeding process, okay, the apical hook formation is takes place. Apical hook formation is takes place. Okay, this apical hook formation, okay, done by the ethylene phytohormone. Apical hook formation done by the ethylene phytohormone, right? Stimulation of radical swelling. Stimulation of radical swelling of stem. We know within the plants there is a radical part is there, right? And this is one of the plumule part is there. The radical part involved in the roots formation. So the radical is basically involved in the root formation. So what it telling here? Swelling of radical, especially that is connected to the stem. We know this is the stem part, and this is the stem part means this is the plumule part. This is the radical part. The radical swelling majorly done by the ethylene phytohormone, right? And horizontal growth of stem. Whatever the plants are there, the plants which are grown horizontally, right? Secondary growth, especially secondary growth. So secondary growth or the horizontal growth majorly influenced by the ethylene phytohormone. So these are the three important points. Okay, these points are very important, and the response is called as triple response. This is related to the ethylene phytohormone. The triple response is related to the ethylene phytohormone. Next one, we will discuss about the abscic acid. Coming to the last phytohormone is the abscic acid. See, the abscic acid, the formula is the C15H20O4, right? C15H20O4, right? That is about the formula for the abscic acid, sometimes asking in bits also, right? See, the very first names of the abscic acid, or in the history, the very first names of the abscic acid, inhibitor B. Okay, first of all, this one is called as inhibitor B. See, we know the ethylene is the one of the growth inhibiting phytohormone. Abscic acid is also growth inhibiting phytohormone. So here, inhibitor B, okay, first name, and next is the abscission 2, right? And next is the dormant 2. So these are the very first names of the abscic acid. Later, whatever the scientists did the research on abscic acid, see, first of all, they concluded this is the main hormone to form the abscic acid. This is the main hormone to form the abscic acid. Sorry, not abscic acid, abscic layer. This is the main hormone to form the abscic layer. See, this is the stem pod. Just imagine this is the maybe the leaf pod or the flower pod or the fruit pod. See, here between these two attachment is there. So where the attachment is there, here there is an abscic layer will be formed. Abscic layer will be formed. That abscic layer formation done by the abscic acid. That's the reason why this phytohormone name given as abscic acid promotes the abscic layer formation, right? And this is the stress hormone. This is the stress hormone. When the unfavorable conditions are there, that time the abscic acid production takes place, and thickening of different plant pods, and the plants are protected from the drought condition and the unfavorable conditions. The plants are protected from the drought conditions and the unfavorable conditions. That's the reason why I mentioned here the stress hormone. This is the stress hormone. During the stress conditions, during the unfavorable conditions, the abscic acid production takes place and protects the different parts of the plants. And this is reported by the Edicoid. Edicoid is the scientist name, or Edicoid, right? The scientist name. Especially he did the research on the cotton fruits, right? And he noticed the abscic acid layer formation and the functions of the abscic acid is identified and named as abscission 2. So here abscission 2 also very first name, right? See, in all these names the very first name is the inhibitor B, next one is the abscission 2, next one is the dormant, and after these three the next name is the abscic acid, right? That is about the history regarding to the abscic acid. So, from which components the abscic acid formation is takes place? Carotenoids. Once the any leaf or any part getting aged, that time carotenoids degradation is takes place. Abscic acid production is takes place, right? And xanthophytes, presence of the lipoxygenase enzyme. When the leaf getting aged or leaf getting senescence, that time the lipoxygenase enzyme act on the xanthophytes, degrades the xanthophyll components, produces the abscic acid. And maverick acid, see maverick acid degradation also here. These are forms the sesamoid terpenes. That sesamoid terpenes, okay, responsible for the abscic acid production. Okay, right? These are the three different components are responsible for the abscic acid formation. And bioacid, flowering and fruit falling test. Flowering and fruit falling test. So we know this one is supporting for the abscic layer formation. Leaves fall out, flowers fall out, fruit fall out, will be takes place. Presence of the abscic acid. And the functions of the abscic acid. These are the functions of the abscic acid. We know the very first function is the abscission layer formation. Once the abscission layer formation is takes place, the water transportation, mineral transportation, food transportation is blocked. Automatically, the particular organ, maybe the leaf, maybe the flower, maybe the fruit, will be followed. That is nothing but abscission. And anti-transparent agent. See, this one is supporting for closing of stomata. Closing of stomata. Some of the phytohormones are responsible for the opening of stomata. In the last class we discussed whatever that the cytokines are there which are responsible for the opening. And see, this is the one of the phytohormone, see, involved in the closing of the stomata. Closing of the stomata, right? That is about the anti-transparent agent, and this is antagonist to cytokinins. Right? Next one is the bud dormancy. Bud dormancy is caused by the abscic acid. Bud dormancy is caused by the abscic acid. See, this one is also antagonist to, okay, gibberellins. Gibberellins are, okay, overcome the dormancy. Gibberellins are supporting to overcome the dormancy. But this one induces the dormancy. This one induces the dormancy, especially buds and leaf senescence. That is nothing but accelerated senescence process. For suppose one leaf, absence of abscic acid, okay, maybe it's having the long lifespan. Once the abscic acid is produced, the lifespan of the leaf will be reduced. Accelerates that senescence stage. Right? And supporting for the flowering process or induces the flowering in certain plants. And the parthenocarpy also influenced by the abscic acid. We know that fruits without seeds. And the rooting process. Rooting. See, some of the functions are present in all the phytoplankton. Suppose rooting. Okay, and the rooting auxins also there. Right. So some are there, but if the options are given, for suppose rooting purpose, auxins are given, and abscic acid also given, then what we have to choose? Definitely auxins. Right? So that is about that functions of the abscic acid. And see, this is a very important point related to that abscic acid. I already said seeds, presence of abscic acid, favorable conditions are there. Presence of favorable conditions supporting for the seed germination. Actually, these are like growth inhibiting hormones, but presence of favorable conditions, presence of abscic acid, the seeds are germinated and involved in the maturation process. But here, unfavorable conditions are there. That time, no germination, no seedling process. So, a single specific phytohormone, that is nothing but the abscic acid, based on the environmental conditions, sometimes it's supporting for, when the favorable conditions are there, that time it's supporting for the seedling process. When the unfavorable conditions are there, that time, okay, it's not supporting for the seedling, or it's not supporting for the germination. Right? That is about the functions of the abscic acid. So that is about the different types of phytohormones. We completed phytohormones. Okay, next one we will discuss about the seed dormancy factors and factors affecting the seed germination right? And next one we'll discuss about that the flower induction process, how the flowers are produced, right? Photoperiodism, that is called as photoperiodism. We'll discuss